Hi everyone, and welcome to Group 18's presentation on epistatic interactions between mutations in the TEM1 beta-lactamase gene. So this presentation will be conducted by Marciana Capetillo, Michelle Lee, Christine Brown, and myself. So to start things off, I'll give a brief overview of what will be presented in this video. Firstly, we will explain concepts from paper number 1, which deals with the evolutionary consequences of drug resistance. Then we will explain concepts from paper number 4, and this deals with the growth rate assays revealing fitness consequences of beta-lactamase. Then we will summarize and explain concepts from an external source paper, which deals with the negative epistasis and evolvability in the TEM1 beta-lactamase. And then finally, we will explain how the external source paper connects to papers number 1 and number 4. Okay. So in paper number 1, which is titled The Evolutionary Consequences of Drug Resistance, the Shared Principles Across Diverse Targets and Organisms, the authors examined various mechanisms of drug resistance in different biological systems. And these systems include the HIV virus, pathogenic bacteria, pathogenic fungi, and even in human biological systems. And the authors also examined the multiple factors that influence the evolutionary trajectory of drug-resistant pathogens. So in our presentation, our focus will be on the E. coli bacteria and the TEM1 beta-lactamase gene that produces an enzyme that confers resistance to second and third cephalosporin antibiotics. So the primary mechanism behind these evolutionary consequences is epistasis. And what epistasis is the interaction of mutated genes which can lower or raise the fitness of a mutant strain. And depending on how these interactions occur, you can have negative epistasis, positive epistasis, sign and reciprocal sign epistasis. And so one detail, well, to get specific, mutations in the genes G238S and R16-4S that encode the enzyme can actually reshape the protein structure depending on the mutation.
Hi, I'm Marciana, and I'm going to be speaking about paper 4, which is growth rate assays reveal fitness consequences of beta-lactamase. So this paper uses the dilemma of antibiotic resistance to study fitness, where they're using the correlation of growth rate assays alongside susceptibility testing to determine which growth rates are more sensitive in testing fitness in bacteria in the presence of different antibiotics. So to do so, they started with the collection of patient isolates, which consisted primarily of E. coli from urinary tract infections, which were resistant to extended spectrum beta-lactamases, which are ESBLs. And UTIs are usually treated with antibiotics, but antibiotic resistance is beginning to limit this treatment against multiple antibiotics and beta-lactamase genes, which are a big contender of resistance genes in the UTIs. But the most common beta-lactamase, which is TEM, which has cost a big chunk of resistance to ampicillin, about 90%, and a multitude of its variants carrying resistance to many other things due to its ability to make conformational changes in its active site, which results in all of these resistant phenotypes. And then after observing these isolates, the most common beta-lactamase, TEM1, along with two others, were identified at higher frequency and higher growth rates. Then, with further comparison of the isolates with and without the TEM1 gene, it was shown that the one without the TEM1 gene had a higher growth rate than those with it, but it's thought that this might be due to the combined effects with CTXM15, which on its own has the highest growth rates. It was also found that a possibility of why the CTXM15 genes have been replaced with TEM genes in bacterial populations might be due to the two genes together resulting in negative sign epistasis when they co-occur, which decreases the efficiency of CTXMs with the presence of the TEM1 enzyme because it hydrolyzes cephalosporins from antibiotics slower. Other observations on growth rates also showed that isolates with the TEM1 gene in the presence of ampicillin had faster growth rates, which was expected as it is a penicillinase. And overall, they were able to find that this sensitivity of growth rates can help show interactions between resistance genes and quantify fitness of bacterial isolates.
Hello, My name is Michelle Lee, and I'll be talking about the external source article that we used to connect the two previously mentioned papers. So this is a screenshot of the title section of the article, and the title of this article is Negative Epistasis and Evolvability in TIM1 Beta-Lactamase, the thin line between an enzyme's conformational freedom and disorder. Okay, there we go. So in summary, the paper aims to explain the molecular mechanisms that cause negative epistasis between two mutations found within the TIM1 beta-lactamase gene. The two mutations are named G238S and R164S. So on the right side, you can see an image of a ribbon diagram of the TIM1 beta-lactamase protein. In this diagram, you can see the positions at which substitutions commonly occur, which are labeled in with a cyan color next to the names of the mutations. The two mutations circled in red are the ones that are studied in this paper. And you can see a red label in the middle of the structure, which represents the protein's active site. So the authors wanted to study the epistatic interactions between G238S and R164S mutations because they wanted to know why combining these two mutations decreases fitness when they are beneficial as individual mutations. In other words, they are researching why does negative epistasis between G238S and R164S inhibit the function of the TEM1 protein, and how does this interaction affect the protein structure? So to conduct this experiment, the authors used a method called hanging drop vapor diffusion, which is defined as a method of protein crystallization where protein solution is suspended on an inverted cover slip over a reservoir. The protein crystallizes within the drop as the reservoir and the solution drop equilibrate to optimal concentrations. So for this study, they crystallized concentrated solutions of TIM1 protein variants, which include the wild type, a protein variant with just the 238S mutation, and another variant with the R164S mutation, and a fourth variant with both mutations combined. First, they placed a drop of a protein solution on the cover slip, and they inverted it on top of a container which contains the reservoir and the precipitant, and the drop stays on the cover slip even though it's inverted. The purpose of doing this is to let the drop and the reservoir both vaporize within the sealed container so that both of their concentrations combined create an environment that is ideal for crystallization. So they did this procedure for all the protein variant solutions, and they collected the crystals to analyze. So what they found for their experiment was that if the TIM1 enzyme has one of the two mutations, either R164S or G238S, the protein structure is altered in a way that allows the enzyme's active site to be exposed. This allows for more space to hydrolyze larger antibiotic molecules in an active binding site, thus increasing the fitness of the bacteria. They also found that if TIM1 enzyme has a combination of both of the mutations, it causes the protein structure to condense. In this case, there is less space to hydrolyze antibiotic molecules in the active binding site, therefore decreasing thickness. So the figure on the right here basically shows what the TIM1 protein structure looks like without the mutation, which is the wild type, and with each of the mutations individually and with both of the mutations combined. The images on the left column show the free form of the structure without any antibiotic molecules binding to it. And the images on the right column show the structure binded to an inhibitor molecule called EC25, which has a similar structure to ampicillin. The purpose of using EC25 was to see how protein structure is affected in the presence of an antibiotic. So as you can see, the mutations individually make the protein structure more open, while the mutations combined make the structure more condensed.
Hi, my name is Christine, and I will be talking about how all the papers mentioned connect. Epistasis is the influence of mutations on phenotypic effect of other mutations, which can be caused by the interactions between mutations, resulting in conformation, stability, or affinity ligand changes. The combinations of the G238 and the R164 mutations take place due to the effect of negative epistasis within the TIM1 beta-lactamase. The TIM1 beta-lactamase is an enzyme that promotes antibiotic resistance, but only occurs as a result of two adaptive mutations. The mutations allow the enzyme's active site to adapt to alternative conformations. and accommodate the new antibiotics that enter the body. These two mutations can lead to a less fit phenotype, negative epistasis effect, through the conformational changes. The TIM1 mutations can be beneficial by changing shape and function of the host and result preventing certain pathogens from biting. But the mutations can also prove to be detrimental to the host when the antibiotics are not able to properly function or fight against invading pathogens. These effects can be seen in epistatic interactions between mutations in the TIM1 gene that affect the fitness course of bacteria and can have major consequences. Mutations increase conformational freedom of the protein structure, but this may alter the accessibility to the active binding site. This means that the TIM1 enzyme cannot bind to antibiotic molecules if they won't fit in the correct active site, therefore decreasing the fitness of the bacteria. Researchers through experimentation have proved that there are little to no compensationary mutations to help compensate for the fitness cost. And now we show the references. These are all the references that we used throughout this presentation, and I want to say thank you for listening.